So several OCA enzyme, that is polynucleotide phosphorylase, was also helpful in polymerizing RNA with defined sequences in a template independent manner. Was also helpful in polymerizing RNA with defined sequence in a template independent manner, that is enzymatic synthesis of RNA. So several OCA enzyme, enzymatic synthesis of RNA, Marshall Nirenberg cell-free system for protein synthesis, and Hargobind Khurana for instrumental in synthesizing RNA molecules, okay, with defined combinations of basis homopolymers and copolymers. Okay, now this is the checkerboard, the codons for the various amino acids. So the silent features of the generic codon are the codon is triplet. Now codons codons code for, and the codon is triplet. 61 codons code for amino acids, and 3 codons do not code for any amino acids. Hence, they function as stop codons. Okay. Now some amino acids are coded by more than one codon. Hence, the code is degenerate. Some amino acids are coded by more than one codon. Hence, the code is degenerate, and the codon is read in mRNA in contiguous fashion. There are no punctuations or commas. The code is nearly universal. For example, from bacteria to human, UU code for phenylalanine. Some exceptions have in mitochondrial codons and in some protozoans. So AUG has dual functions for methionine and also act as initiator codon. So UAA, UAG, and UGA. They act as stop terminator codons, right? If following is the sequence of nucleotides in mRNA, predict the sequence of amino acid coded by it. So AUG is for methionine, then UUU is phenylalanine, then UUC phenylalanine, UUC, UUU, all these are phenylalanine. Okay. Now mutations and genetic code. Now a classic example of mutation, point mutation is a change of single base pair. In the gene for beta globin chain that results in the change of amino acid residue glutamate to valine GAG to GUG. Now it results into a disease condition called sickle cell anemia. Okay. Okay, so insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. So such mutations are frame shift insertion or deletion mutations, right? Now insertion or deletion of three or its multiple bases insert or delete in one or multiple codon. Hence, one or multiple amino acids and the reading frame remains unaltered from that point onwards. Insertion or deletion of three or its multiple bases insert or delete in one or multiple codon. Hence one or multiple amino acids and the reading frame remains unaltered from that point onwards. Okay. So it's like three bases. If you delete it, it remains the same. Okay. Now tRNA, the adapter molecule by Francis Crick, and no structural speciality to read the code uniquely. Okay. It is adapter molecule, and also known as soluble RNA, sRNA, and it has anticodon loop that has bases complementary to the codon on mRNA, and it also has an amino acid acceptor end, CCAN, to which binds to amino acids. tRNAs, these are specific for each amino acid, and for initiation there is initiated tRNA. No tRNAs for stop codons, and secondary structure tRNA is clover leaf, and actual structure it is inverted L. So actual structure inverted L, and second structure is clover leaf, as you see in here, like 5-3 5-3 tRNA, serine, tyrosine. AGU has codon. Anticodon would be UCA, right, and UAC, so AUG, right, mRNA, so 5-3 right. Then in the translation, right. Now translation refers to the process of polymerization of amino acids to form a polypeptide. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. There is peptide bond formation, and it is energy requiring process. So therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their conjugate tRNA process commonly called as charging of tRNA or amino acylation of tRNA. To be more specific, okay. Now also there is catalyst that can enhance the peptide bond formation rate. Now cellular factor is responsible for synthesizing proteins in the ribosomes. Okay, the cellular factor is responsible for synthesizing proteins is the ribosomes, right? Now, ribosome consists of structural RNAs and about 80 different proteins. So, in its inactive state, it exists at two subunits, a large subunit and small subunit. So when small subunit encounters an mRNA, the process of translation of mRNA to protein begins. Okay, there are two sites in the large subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of peptide bond. So, ribosome. Now, the ribosome, they also act as catalyst. That is 23 srRNA in bacteria is the enzyme. No, 23 srRNA, it is a structural RNA, and this ribosomes, like, it's an enzyme, okay, and it's a ribozyme, so for the formation of peptide bond, so it acts as an enzyme. Now the translational unit in mRNA is the sequence of RNA that is flanked by a start codon, that is and stop codon, and codes for a polypeptide. So an mRNA has also some additional sequences that are not translated and are referred to as untranslated regions, right? UTR, and UTRs are present at both ends, like before the start codon, that is 5 end, and after the stop codon, that is 3 end. So they are required for efficient translation process, right? The ribosomes moves from codon to codon along the mRNA. Okay, the amino acids are added one by one, translated into polypeptide sequence detected by RNA, detected by DNA, and represented by mRNA. So amino acids are coded one by one, added by one, and translated into a polypeptide, a complete polypeptide. Now sequence, amino acid sequence is detected by the DNA and represented by the mRNA. So at the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosomes. So first one is like recognition of the initiated tRNA, and then elongation, right, and then release, that is terminating. So the regulation of gene expression.